Yeah, but she arrived at 6:05 or something like that. Okay.
A general overview of the different tools that have been derived from viruses. And you will see that it's quite a broad topic. And of course, a lot of molecular tools are derived from viruses, and I will explain some of them. Now, viruses are used as expression vector in cell culture. They're used for gene therapy in vivo. We use them as biological control agent. And I'm going to describe some surprising example, mainly from Australia. And I want to stress once again that I have no problem with Australia at all. It's not my fault that they did surprising things in the past and still at the moment. And at the end, I will address the use of viruses as substrate of vaccine development. And as I promised to the Francqui share, I will try to give advice to the young generation throughout these different examples. So let's start with the tool. In your tools of molecular biologists, you have a lot of different reagents that you are using every day. And some of them are derived from viruses or from antiviral immunity. And this is something that I'd like to illustrate with two first examples that you use every day in the lab. And surprisingly, very few people knows exactly that this is related to viruses. So the first obvious example is restriction endonucleases. Use them every day. I did an experiment last week. I asked a few people in my lab to say, what's the origin of restriction endonucleases? And people say, oh, it's Promega or Rogentech. I say, no. What's the organism that produced them? And few people replied, it must be biochemist. I say, no, what's the biological entity from which it has been cloned? And surprisingly, a lot of people ignored that it has been derived from bacteria. And what is even more surprising, when you raise the question, what's the purpose, what's the biological meaning of restriction enzymes in bacteria, very few people are able to explain that these enzymes are part of the innate immunity of bacteria. And that's the purpose of restriction enzymes. If a phage tries to infect this bacteria, and if this bacteria express a restriction endonuclease that has the ability to cut the genome of the phage, then the infection is blocked. And that's the biological meaning of these restriction enzymes. They've not been produced by nature to please molecular biologists. They are part of the innate immune system of bacteria. And these three guys, this is, in my point of view, one of the most coherent Nobel Prize. Arbert discovered the restriction enzymes. Smith identified this palindromic sequence that are recognized by the enzymes. And Nathan's demonstrate that you can use them to make something that you probably don't know what is a restriction map. So restriction map is basically a characterization of the restriction fragment of a genome. Nowadays, it's become ridiculous to do so because you sequence an entire genome in just a few weeks. While I was preparing this talk, I remember that my first paper in my CV is actually called Restriction Maps of Cervin herpes virus 1 and Cervin herpes virus 2. And it took me 4 years to complete it. So this is something quite frustrating in science, is that the progress of the techniques is so fast that if you look at what you did at the beginning of your life, it becomes ridiculous. But altogether, it's a beautiful system. Another system that you use every day now is CRISPR-Cas9. This is also part of the innate immune system of bacteria. At the origin, this is the purpose. And this is actually a beautiful system that in terms of immunology, if you have to classify it, it's very embarrassing. Of course, we know that adaptive immunity appear in the vertebrates and only vertebrates that have a jolt. The lamprey, for example, does not have an adaptive immune response. But if you look at the system of the CRISPR-Cas9, and I will explain it in a minute, you cannot say it's a pure innate immune system, because an innate immune system, by definition, has no memory. It's not able to take into account and to learn and to improve by collecting a trace from the past. And the CRISPR-Cas9 is actually recording some experience in the past. And this is how it works. So you have this Cas genes and the CRISPR locus that will basically collect throughout evolution sequence of the phage that try or succeed to infect this bacteria. And then using this CRISPR locus transcription unit, it will produce a pre-RNA that will then be cut, you know that, and this will serve as a guide to hybridize with the genome of the phage, and thanks to the Cas9, the genome will be cut. So that's a very, very smart system that allows the bacteria to learn and to remember the phage it faced to. But more importantly, this system can be transmitted from generation to generation. And this is even more bright than all adaptive immune system, because all adaptive immune system, you cannot transmit it to the next generation. While this system here, a bacteria can pass to the daughter cell the information of previous infection and transmit away a mechanism to protect them. So that's again, something that has been discovered, not studying virus, but the antivirus immune response, and that we use every day. It's an extremely extraordinary system. And we use in our lab in zebrafish, for example, and you can do homozygous KO by simply injecting one-day embryo. It's extremely powerful system to make KO, but now increasingly to make K-in as well. So very good system. Not surprisingly, this discovery of the system is, was rewarded by the Nobel Prize last year to Emmanuel Charpentier and Jennifer Doudna. Okay, what are all the tools, tools that you use as a molecular biologist? Well, if you just analyze a simple plasmid, you will find a lot of different sequences derived from viruses, promoters, CMV promoters that you find in 50% of the expression plasmid. This is a promoter cloned from human cytomegalovirus. Origin of replication, the FD40. This is the simian virus 40 origin of replication that you find into a plasmid. It's a sequence that has been cloned from a virus to put into this plasmid to make it able to replicate in eukaryotic cells. SV40 polyadenylation signal, etc., etc. These are all pieces of sequences that have been cloned from viruses. More interestingly, IRES, IRES, and again, if I ask you what's an IRES, you will all answer it's an internal ribosome entry site. Then if I ask you where is it coming from, it's more difficult. And it's actually a sequence that has been identified in 1888 by Sonnenberg as a site where ribosome can be recruited in a cap-independent manner. And this is extremely smart for a virus to acquire this system, because the innate immune system of a cell, what we call cell autonomous immunity, is able to detect quite rapidly that the cell is infected. So a cell completely isolated can sense and detect its own infection by a virus. And when it does so, it will immediately activate a different cascade pathway that will lead to a blockage of a protein synthesis. And this is very, very embarrassing for a virus, because when you are in a cell and the cell is now refusing to translate your protein, you cannot replicate. And all these recruitment of ribosome are done in a cap-dependent manner. And so some viruses, not surprisingly, acquire through evolution these special RNA structure that allow them to recruit ribosome in a completely independent, cap-independent manner. And so they can continue to produce their protein in a cell that has been informed of the infection, a cell that has taken action to block protein translation. And so that's the biological meaning of IRES. It's not to make one polycistronic RNA. We use them for that, but that's not the initial purpose. The purpose of the virus which acquire these sequences, and you find them in different viruses infecting animals, but also infecting plants, this structure is to make translation independent of cap. Of course, we use them in our lab to make polycistronic mRNA that with one mRNA will encode different proteins, but that's not the evolutionary meaning of this IRS sequence. I promised last week to talk about T2A peptides. So in construction, it's quite frequent that you want to make two protein encoded by the same mRNA for space reason or stoichiometry or whatever. And one trick that is frequently described is the 2A peptides. So what are these 2A peptides? These are sequences that are found in viruses. Again, it's viruses that have acquired this system. And it's an 18 to 22 amino acid sequence. And you have here, for example, the alignment of different 2A peptides. The first letter stands for the virus from which it has been characterized. So P is for porcine, T is Tezoa assignata. It's a virus infecting butterfly, and E I don't remember. But when you align them, you see that the end of the peptide is conserved from all these viruses. So what will happen? If you make a cloning, so you take the sequence of a first protein, so they call it gene, it's not correct, it should be open reading frame. So you take the sequence of a first protein, then you make in a single open reading frame the sequence coding for the peptide. Let's say we use T2A, then the sequence of your second protein. So if you take this sequence, you will in fact have a fusion protein consisting of protein 1, the 2A peptide, and protein number 2. Now, this is a single open reading frame controlled by a single promoter, and you will produce an mRNA. When the ribosome will translate this mRNA, three situations will occur. The first one, and this is what you hope, when the ribosome is facing the end of the T2A, the 2A peptide here, it get a problem to make a link between the G and the P amino acid. And as a consequence, frequently, it interrupts the translation. So meaning that it translates the protein and then it has a problem of structure, and then continue, but there is no link between these two amino acids. With the consequence that at the end, you produce two proteins out of one mRNA. But keep in mind that your first protein will keep the tag that represents the major part of the 2A peptide. And your second protein, rather than starting with a methionine, will start with a proline. This is what you hope. Now, what the system doesn't tell you, but which is the reality, is that this is what you look for, but in fact, the ribosome will, quite frequently, will also drop off the mRNA. So releasing your first protein with the tag, but without synthesis of the second protein. And the third case, that sometimes the ribosome will manage to make the link between the G and the P and will basically produce a fusion protein. So when you use these two T or whatever 2A peptides, you have to be careful and keep in mind that you will not produce just your two protein, but in fact a mixture of the fusion protein, your first protein alone, or the two protein of interest. But in all cases, you will end up with a foreign sequence, which is the tag present at the end of your first protein or a proline at the beginning of your second protein. So in contrast to what is described in textbook like self-cleavable peptide, it's not a self-cleavable peptide. It's just a way to block and interrupt translation, but it's not cleavage. That's very important. There's a quite nice review in Scientific Reports a few years ago, but quite good, where they compare the different 2A peptides and even use them in combination. Everything okay? I told you last week that we produced an interesting T2A GFP cassette, and I give it for free. So if you need it, I can pass it to you. It's a funny cassette. It consists to the human elongation factor 1 alpha promoter. So it's a human promoter, but which surprisingly works in a very broad range of animals. You can even use it in oysters, in eels, in carp, in chimpanzee, whatever. It works in every single cell that we try. And then you have a single open reading frame, which encodes for lecture2. So this is a luciferase. Then you have a T2A peptide that has been derived from a virus infecting a butterfly. And finally, you have the copper-pod GFP. And now when you do this, you put this cassette into a virus. This is what we did here. Ayan Zhang is a Chinese PhD in our lab, and she produced angulate herpes virus 1, which encode this cassette. And as a consequence, this virus is bioluminescent. You can take the ill and put it in an IBIS machine and see the site of inoculation. But if you take the specimen and go to confocal, you will see the cells, because the cells are simultaneously bioluminescent and fluorescent. So this is very, very, very efficient technique. Now, I think science is sometimes missing some philosophical thought. And I think from time to time, you should realize that you are, well, we are all privileged scientists. But if you think about Pasteur, for example, the guy has a culture medium that he was preparing like a soup. He had glass, a flame, and that's all, and an incubator. He had no tools at all. And your generation, you have amazing tools available. And more importantly, you really have the whoops, Dirk is doing experiment again. You have the possibility to create something. You dream of something, you draw it on paper on your computer, and in a week or two you can produce it. And this is exactly what has been done here. If you think you have this lecture2 coming from firefly, the promoter coming from human, you have the T2A, as I mentioned, coming from this virus infecting a butterfly, and then you have the copper-pod GFP. This is a GFP which is much stable than the classical eGFP. And it has a great advantage that it works from 4 degrees to 42 degrees. So if you have to do eGFP experiments that are not in the classical range of temperature, for sure, that's a good option. Yeah, so that's, when you look at this construction, you must sit down for a minute and say, okay, that's amazing. We create something. It's really, I don't like creationism, but you reach the stage where you can create something. You can create a protein, a biological entity that did not exist. And once again, I think in science, it's good to sit down and to think about what you're doing. And I advise you, if you go to Roma, to visit the Sistine Chapel. You have to prepare yourself. It was expecting it's huge, but very large, and actually it's very small. And they have this famous painting of Michelangelo where God is creating or giving the power of man to create something, and that's what we're doing in science, and we should respect this. And when we're drawing these sequences, I think it deserves some time to sit down and think of what we're doing, and to appreciate that we have this privilege. A man like me, I'm 54, and I end my PhD thesis without doing a single PCR. And the same lifespan, you can now draw something on a piece of paper and order the primer on the Monday to make it in a couple of weeks. So we really living a great scientific experience. Other example of proteins that are in our toolbox and that we use every day. If you want to make a cDNA, you know that you use a kit, and that's another thing that as a young scientist, you are very efficient, because you have the shelves are full of biology of kits. But please try to understand and to learn, to be a little bit curious of what is in the kit. And in the kit to do a cDNA, well, there is a very special enzyme, which is basically the reverse transcriptase. And this is an enzyme that has been cloned from virus, because retroviruses, as you know, that the genome consists of RNA And these viruses integrate their genome into the host genome. And to integrate a single-strand RNA molecule into a double-strand DNA molecule, you obviously need to transform your RNA into a double-strand DNA. And this is what this enzyme is doing. And it's actually three enzymatic activity. You start with RNA, then you make a heteroduplex with an RNA-dependent DNA polymerase activity. Then this heteroduplex, of course, it's not a double-strand DNA, so you need to get rid of the RNA, and you need the RNase activity that works on heteroduplex. That's not trivial. There are not many RNAs that are able to cut an heteroduplex. This enzyme is able to do so. You remove the RNA, and then finally you synthesize the complementary strand of DNA. And for this, you need a DNA-dependent DNA polymerase activity. So all these enzymatic activity have been cloned from these retroviruses, and this is what you found in this kit. Other example, LOXP. If I ask people where are LOXP sequences and Cray recombinase coming from, again they say Promega, England Biolabs. No, it has been cloned from viruses. This has been cloned from bacteriophage P1, temperate bacteriophage that insert the genome into the genome of the host. And this is a fantastic system that we nowadays use in molecular biology for different purposes. Just a brief description, but if you take two LOXP sequence, will have different effect based on the number of LOX sequence that you put and the orientation. If you flank a gene with two identical LOXP sequence, but in opposite direction, and you induce the Cray, you will switch the orientation of your gene, which can be very useful to switch on or off based on the promoter that you put on the left side. If the two sequences are flanking in the same orientation, you will induce a deletion. And you know it, this is what you use to knock out a gene after a development. If an animal cannot be developed without a gene, then you flux it. Then when the animal is fully mature, you induce Cray expression that will genetically remove the sequence from the gene. You can also use it for translocation. So these are all kind of tools that have been produced based on molecules that have been characterized in viruses. And I could continue like this up to tomorrow, until tomorrow. They are full of examples. Okay, doc. Second point, viruses as expression vector in cell culture. Well, I already addressed this when I described baculoviruses. So baculoviruses are, in my point of view, one of the smartest viruses. So they produce this virion, which is fully infectious. But more interestingly, in the infected cells will produce huge amounts of a protein called polyhedrin. And this polyhedrin will, well, it's a kind of crystal, will assemble around the particles, the virions, so that when the infected cells die, it will basically release what we call occlusion bodies, virions that are trapped in a matrix of polyhedrin. So what's the purpose of that? Well, it's very important for the biological cycle of the virus. This is the drawing of a caterpillar. I show you a picture during the first lecture. When the caterpillar is infected by the baculovirus, it basically turns into liquid that leak to the leaf of the plant. And the exudate, the liquid of the caterpillar, contaminate the leaf, releasing the occlusion bodies on the leaf of a plant. Now, for a virus to be on the leaf of a plant, it's not a good idea because, as you know, there is the sun, there is the rain, etc. But thanks to these occlusion bodies, the virions contained into these occlusion bodies will be protected from UV light. And so the double-strand DNA of this virus will not be affected because it's protected from the polyhedrin. Now, it's good to be protected, but at some stage you need to be released to infect another naive host. So this is, again, nature is well done. When a young caterpillar will eat the leaf, which is contaminated with these occlusion bodies, well, these occlusion bodies are digested in the digestive tract of the caterpillar, releasing the virions that are then now able to infect the new caterpillar. So beautiful system. where a virus has learned to unwrap, to pack its variant to protect them and make them lasting for days, weeks on the leaf of a plant rather than minutes without this protein. Now, viruses are smart, but scientists too. And one thing that we always do in science when we want to address the role of a protein or a component, and you can do it in life too, is to make the system working without this component. That's a very, if you want to know if one person is important in your life, try to live without this person. If you live perfectly well without this person, it means that you don't like her or him. So virus and gene is the same. If you think a gene is important for a virus, make a virus that has been deleted for this gene. If the virus replicates normally, then it means that this gene is not imported into that system. So scientists look at the gene for polyhedrin and they remove this gene from the virus. And they observe that the virus replicates in cell culture perfectly well, simply because in cell culture, the virus does not need to be protected from the sun, from the rain, etc. So this was interesting. Then other scientists saw, well, that's very interesting. If this virus produces a lot of polyhedrin, it means that it has a very strong promoter driving this open reading frame. And if the virus can survive without it, that means that we can replace the synthesis of polyhedrin by a protein of interest. And this is where some people try to take a protein of interest and clone it into this polyhedrin locus to make the virus producing the protein of interest in place of polyhedrin. And this was the beginning of a new era of using baculoviruses as an expression vector to produce protein in very, very large amount. So the system is very simple. Pharmaceutical companies produce huge amount of these insect cells called SF9, the Spodoptera frugiperda clone 9, and you amplify your cells. And then when you have enough cells, you inoculate your cells with your recombinant baculovirus that will infect the cells, produce its own protein and produce variants, but in addition will produce the recombinant protein of interest. And you will, if you analyze the wild-type virus on the left and the recombinant baculovirus on the right, you will see that the polyhedrin will disappear and will be replaced by the recombinant. Now, what very few people know is that the Cervarix, and some of you may have been vaccinated with this, it's produced like this. So the antigen of the virus-like particles is produced in SF9 insect cell. So very, very useful system. Production expression of protein using viruses. Well, there are, again, hundreds of examples. You can use it in a transient way, like I just described. You take fresh cells, you infect with a virus, and then you have a one round of expression and the cell dies. You can also use viruses to stably transform cells. And this is typically of lentiviral vector, where you clone and make a construction into your virus. Then when these cells will be infected, you will have an integration, random integration, fortunately, into the genome of the host. And if your construction consists of an expression cassette inducing the expression of protein, you will end up with the expression, stable expression of the protein of interest. But you can also use it to switch off the expression of an endogenous protein if you use the lentivirus to induce the expression of the silencing RNA. So there are really a very, very large possibility of combination to make transient or stable expression using viruses. Viruses as tools for gene therapy. Well, this is a very important issue. And I'd like to, first of all, to stress the fact that every single thing that we do, and it can be a medical intervention or even a practice in nature, must address the safety and the efficacy of this measure. So the safety is it safe for the subject, for the environment? By doing this, do we expose the subject or the environment to negative effect? And it can be extremely diverse, as I'm going to show you later with surprising examples. And then the second aspect is the efficacy. is it a good way to reach my goal? Always ask yourself, where do I want to go and how to reach it? But if you don't expect an efficacy, then don't expose your system to a potential risk, even low. And this is something that we must obsess you as a scientist when you propose a measure to solve a problem. And it's certainly the case when you talk about gene therapy. In gene therapy, you can basically classify them in different ways. And one is certainly what we call in vivo and ex vivo. And you will see that this is related to the safety of the approach. So in vivo, you will inject your virus on which you bet and you hope that it will genetically transform your defect. But this virus will be injected straight away into the host. That's amazing. You take a virus, genetically modified, you inject it straight into the patient, sometimes using specific site of injection, but you really drop your virus into the host. So you can expect that you can only use extremely safe virus to do this approach. And this is called the in vivo. Then another approach is called the ex vivo. Basically, you take the cell from a patient, because we all have MHC class 1 and MHC class 2 that are different from individuals. So this is a one-by-one treatment. And this is why it's so expensive. So it's a case per case situation. You take the cell of one patient and you will transform these cells ex vivo. This is, of course, much more an interesting approach in terms of safety, but also in terms of efficacy, because if you imagine the multiplicity of infection, when you drop virus into a host, the multiplicity of infection is very, very low. There are a lot of cells into a host. While in a petri dish, you can control the amount of virus that you put because the cells are much less abundant. You can also adopt some selection process and control that the modification you want has been done. And then finally, you re-inject these modified cells into the patient. So that's this real classification in vivo, ex vivo. Now, in terms of viruses in association with these two main concepts, in vivo, ex vivo, they're actually the parallel of two main types of modification. And this is relying on the fact that the virus that you use to induce the modification to restore the gene will integrate or not into the genome. And as you can expect, viruses that are integrating the cassette into the genome will only be used ex vivo, while the viruses that are safer and do not insert their genome into the host genome will be used both ex vivo and in vivo. And finally, you have the non-viral vector unit. Now, this is one table that I added into the slides. It's a kind of comparison of the four main type of virus vectors that I use for gene therapy. And you can clearly see that some features are exclusive. For example, if you look here, the integration, these two are not integrating into the genome, and they can be used in vivo. While those ones that integrate can only be used ex vivo. It makes sense. It's again a kind of safety efficacy issue. A key feature also when you think about gene therapy is the packaging capacity. So the length of the genetic material you can inject into the genome of the virus. This is frequently a limiting factor, and you see that nowadays we have, especially with adenoviruses, a quite large broad range of insertion, 36 kb. This is huge, but in most cases you are around 5 to 10 kb. Yeah. A key issue, if you want to make in vivo, is certainly the tropism, to send the information to the right cells. And this is in virology a very important issue. And that's actually something very, very important for a virus. A virus has an address on its surface. When it goes in, he has an address to go to particular cells. When he goes out of the host, he must have another address, otherwise keep going to the same cell. So this is a fascinating aspect of virology, but it's certainly also something fascinating for people who are addressing gene therapy, because you can use the different tropism of the different viruses to target different cells. But nowadays, due to the fact that we can create new viruses, you can also make a virus that has a new address or change the address to drive your virus to specific cell. And this is, I think, something that is really fascinating in this topic. Just to give you an idea, if you look at nowadays at the number of clinical trials in gene therapy, viruses represent about 50% of the trials. And they dominate by adenovirus, followed by retro-lenti, ex vivo, of course, and then the adeno-associated virus. So if you want to have a future in science in this topic, for sure, there are opportunities. Yeah, that's, again, about the creationism. You know that we have in our toolbox as immunologists, you have, for example, single chain antibodies that you can produce by linking a variable domain of a heavy chain and a light chain, or nanobodies that you develop in alpaca. And you can use these molecules specific for a defined epitope, for example, to build a virus that will have this address on its surface. So nowadays, we can really design viruses that will target specifically one cell expressing a particular epitope. So this is not science fiction. This is what we're doing now in immunology and virology. Most people think that gene therapy is still something for the future. Well, that's not true. Huh? There are treatments on the market nowadays. The first FDA-approved gene therapy was for this rare retinal disease. I most probably remember this visual cycle. I tried to remember yesterday when I was preparing this talk. I studied this in the first year in the biochemistry course. It's the retinal, then it becomes trans-cis. You remember this, huh? With the pigmented. You don't study this nowadays, no? No, you completely forgot. Yeah, good. No scars in your brain about this. I remember studying it. And there is one protein here called RPE65, which is essential to make this cycle turning. And if this enzyme here, this protein is broken, basically you don't produce the pigment into this pigment epithelium layer here, and you become blind quite rapidly. So you're born and you see, but because you don't protect your epithelium with the production of this pigment, very, very early people become blind. And so some scientists had the idea to develop a gene therapy for people known to carry this autosomal mutation, and then to basically restore the synthesis of this protein in the cell by injecting adenovirus expressing this protein. And this has been designed, it's approved by the FDA, and it has been very recently approved also in Europe. So these people who unfortunately are genetically mutated and have a defect, the surgery consists to take a suspension of adenovirus expressing the normal RPE65 protein, and then the surgeon will inject this protein here in so below the neurons that are here, will inject it to the retinal pigmented epithelium, and this will lead to the expression in these cells of a normal RPE65 protein. So it does not solve the disease of these people, but it makes the destruction of the epithelium much slower, and the results are quite impressive. So you don't have an insertion here; it's an episomal form of the adenovirus that will be remain as an herpes virus latent. Will be an episome close to the chromosome of the host cell. So you don't have integration. So procedure is quite, well, it must be quite stressful that you inject this into this, don't drink too much Diet Coke before doing the injection. It's quite frightening. viruses as biocontrol agent. That's a funny part. Biocontrol means that you use a biological entity to control another biological entity, right? That's the principle of biocontrol. So how can you do biocontrol? Well, we're using virus. Well, I will discriminate two situations. The first, you can use a virus to kill a target host species into an ecosystem, right? Let's imagine that you want to kill rabbits. You don't like rabbits. You had a problem with a rabbit when you were a small boy, a small girl. You want to get rid of rabbits. You take a virus killing rabbit and you spread this virus into the ecosystem. This virus, if it is specific for rabbit, it will kill the rabbit and don't kill the other species. There is a more complex situation that I will address also, is to use viruses to kill target cells into an organism. So here, you want to destroy specific cells into an organism which is also composed of cells. And the cells that you want to kill, there are two possibilities. Either they are cells, so meaning cells derived from this organism. So what type of cells do you want to destroy in an organism? Well, for example, tumor cells. If you have a tumor inside your body, it would be interesting to have a virus that kills them. This is called viral oncolysis. So you use the virus to kill abnormal cells in the organism. You can also use viruses to kill non-self cells into the organism. For example, if you are infected with bacteria, you could use bacteriophage to kill the bacteria into the host. And this I will explain also. Now, this is very important. I already stressed the importance of safety and efficacy, but I'd like to come back on this. So in terms of safety, if you use a virus to kill a target species in the ecosystem or cell into an organism, one obvious aspect of the safety is that your virus kills what you want to kill and does not kill the others. If you distribute a virus that killed the rabbit, but it kills also the squirrels, young boys, etc., it's not useful. You want to have something highly specific. And this is quite obvious in terms of safety. But if you think a little bit more, there are other aspects of safety that are concerned by the use of viruses as biocontrol agent. When I describe the evolution of viruses, I describe two main type of evolution. There is the co-speciation process, and then there is something more embarrassing, which is the host jump. Sometimes viruses jump from one species and land into a non-natural species where they are able to replicate and create a disease. One aspect of using viruses as a biocontrol agent is that you will promote host jump. You will take a virus and you will expose it, produce it massively. You remember this picture I show you of the plane spreading baculovirus on the field. You expose the ecosystem to a huge amount of virus. And so you create opportunities for a virus to jump into a species that is not a natural species. And so one aspect of biocontrol, the use of viruses as biocontrol agent, is that we create by this process opportunities for host jump to occur. And so we have to be extremely sure that and control this experimentally, that this host jump is not possible. Now, it's also an issue about efficacy. You remember that I explained that the viruses will act as a pressure of selection on the host. So meaning that if you take a virus, drop it in the ecosystem, and it kills 99% of the rabbit, but does not kill all of them, your virus will have an effect on the host diversity. It will select the resistant one, but as a consequence, your host will be resistant, and it will have an effect on the pathogen, and you will lose your efficacy. So before using this type of tools, you should think, is there any possibility to select a host which is resistant to my agent, and second, how fast these resistant individual can repopulate as the system. If you kill bacteria and you kill 99% of them, but the 1% survive is fully resistant, the next day, the bacteria are back in business because they replicate every 20 minutes. It's okay if you kill humans because humans takes a while to replicate and they don't make a lot of offspring. But if you talk about a species that is highly prolific, that has a very short generation time, then this has a massive impact on the efficacy of your system. And you can lose the efficacy on your system very, very quickly in few years or even few months. So this is very important to keep in mind, these two aspects about safety and efficacy that both rely on this interaction and evolution of the pathogen, but also the host. Very, very important. Okay, so some example of a biocontrol agent, baculovirus, I mentioned it many times, this virus that have a very, very narrow host range. That's very good. They kill one butterfly, but they don't kill butterflies that are few million years ago, few million years away. So it's very a surgical virus that will kill one target species, but not the other one. And I show you this cycle many times where you will basically maintain in your system. You actually spread once the virus, and then your the virus that will be released by the caterpillar on the leaf will in a way immunize the plant from reinfestation, because the next generation of caterpillar that comes eat the virus produced by the first generation and die. So it's very, very efficient. And in the United States, it's now massively used and has a very, very good impact on nature, because previously they were using pesticides that had a dramatic effect. They were killing all the insects, but had also impact on the birds. You remember this sad story about the egg shelf of the birds completely damaged because the birds were intoxicated by the DDT. So thanks to this modern approach of biocontrol, well, it has a positive impact on nature. Viruses as a biocontrol measure against rabbit. So again, I have nothing against Australia. Now you will see that I will charge them a little bit, but it's not their fault that some people do stupid things on their content. So the rabbit, so the European rabbit is not an animal, natural animal from Australia. It has been brought there a while ago. I think it was in 1788, initially as a domestic animal. People were produced them in farm. And then some people thought, well, it's fun hunting rabbits. Oh, I don't think so, but anyway. They thought, let's release rabbit into the wild so that we will have what they call game farming. We will invite our friends to shoot the rabbit. And they did so. And this turned out to be a good idea, but for a short period of time, because, you know, rabbit proliferate like rabbit. Huh? It's not a fake news. They replicate like mad. And very rapidly, in about 40 years, the situation went completely out of control. And then they just say, okay, but let's develop tourism. Huh? We are at the beginning of the 19th century. Let's make fun and game farming. And you see, this is an advertisement of tourist company is of that time, and they promise people to shoot at least 400 rabbit per hunter per day. That's one. So these guys had to have different guns, because otherwise it come too hot, and you put the cartridges in, and the cartridge blew up. So this amazing scene where the guy had 5-6 guns and people just putting in the cartridges and the guy shooting. And this is one afternoon session. The truck is full of rabbit all around the rabbit. Okay, so this was good, but the rabbit dig holes, and when they overpopulate an ecosystem, they eat everything. They can even kill trees because they bite the skin of the trees. And not surprisingly, it turns out to be a disaster, because when you have the heavy rain season in Australia, if you don't have roots that maintain the ground, you have this kind of disaster. So very rapidly, people realize that these rabbits becoming a pest. And this is the kind of scene that you see, it's a desert because they eat even tiny piece of grass, they take it and it turns out to be a desert. So they thought, well, we need to control these rabbit population, and they opened at the end of the 19th century a world competition to develop a measure to kill the rabbit. And actually, Louis Pasteur, at the end of his life, submit an application to this competition. And I'm still trying to find the agent he proposed, but he participated to this competition. Then a guy came with a very, very bright idea. He had the idea to build a fence to prevent the rabbit from spreading into the field. And this is very, very surprising. So how can a scientist propose to build a fence to prevent the spreading of rabbit? If you ask my father, he didn't went to the high school, he's a farmer, but if you ask my father, will a fence prevent rabbit from progressing into the field? He will laugh. He will laugh like mad because he knows that rabbit dig hole, right? This guy, this government, advised by top scientists, didn't know that rabbit were digging holes. And they took the decision to build a fence of several thousand kilometers. Can you believe it? And this is the fence they create, going north to south in Australia. And Australia is not a small country like us, our. That's the first fence they built, fence number one. They turned out to be completely useless. Then, just to make sure that it was not a mistake, they built the second one here, and then they built the third one. Unbelievable, completely stupid. At least it makes the rabbit laughing. And this is so they start building this fence in 1909. And you see here, it's called Mr. Stevenson suggests that the government should create a wire fence along our New South Wales border in order to check the coming invasion of rabbit. The artists depict the probable use the bunnies will make of the fence. So they use it to play tennis. So of course it was completely useless. And remember what I told you, safety, efficacy. In terms of efficacy, this is useless. So probably one of you politicians has a company building fence, or I don't know, but it's completely irrational. Now think about safety. This procedure, efficacy, 0. Safety, could it be negative or not? You say no, it's a disaster. Because this region of Australia is the source of migration of plenty of animals. They cannot fly, they cannot dig hole, like the emu. Yeah, but this is not funny, because the emu were completely surprised and get in front of this fence and say, what shall we do? We're supposed to migrate from east to west and west to east. And now these monkeys built the fence across our country. And so each time you develop a measure, even building a fence, you must, as a scientist, think safety and efficacy. And safety is not a stupid system. Oh, by the way, competition for champagne is finished, but I think you realize that I like going to the movie, and there is a very, very beautiful movie. I don't know if you know it, Rabbit Proof Fence. No? It's difficult not to cry when you watch this movie. And it's actually unfortunately a real story. But at some stage, the British thought that the children of Aborigine deserved a better education. So they did something completely horrible. They were taking the children from their own family and make forced adoption to clean British that were coming there. And this is the story of these 3 young ladies here. And they were living, I don't remember if it was south or north, but close to the rabbit fence. And they were export at the other side of the country. And they realized one day that the rabbit fence was close to the new place. And so they knew that if they were walking along these fence, one day or another, they will reach their own place and recognize their place. And the movie is the story of their journey of 1,500 miles. So these two, because the two, they were separated from the small one, but these two walk, and this is a real story, they walk along the fence for more than 2,000 kilometers and finally found their place. And it's revealed how these these young girls were able to survive in this completely wild and rude environment. And it's very, very nice. A very nice movie. Now, another story came. The fence turned out to be a stupid thing. So they said, let's try to find something that killed the rabbit and not the other species. And at the other side of the world, in Brazil, some European went there with European rabbit. And they realized that the European rabbit that were brought in Brazil were dying from a disease. And the agent was identified as a virus called myxomatosis. And this virus does not cause any lethal disease in its natural host because it's well adapted there. So the Brazilian tapiti, sometimes it has benign lesions, but which will vanish in just a few days. But when the same virus is inoculated to all rabbit, the European rabbit, then it developed this horrible disease that you may have seen in the forest. It is a blind rabbit with myxoma everywhere. And this is actually a beautiful illustration of the co-speciation process. You have a virus that is now fully adapted to its natural host, but if this virus makes a host jump into this species, which is not far away from this one, is completely non-adapted and create this horrible lethal disease. So people thought, let's try to use myxomatosis to kill European rabbit into Australia. And so they started an experiment on the 16th November 1937, and well, they were a little bit concerned about the biosecurity. So they released infected rabbit on the Wargdan Island, source of Africa. It worked perfectly well. Rabbits were dying like flies. And so they say, wow, that's it. We found the trick. And then before they took the decision to release the virus on the main continent, they realized that the virus was there. So the virus basically jumped from, so safety was totally, the confinement was completely a failure. And so what they did, these guys, not to be in trouble, they declared that this was a very good trick and they should release the virus immediately on the main continent. But the virus was already there. So they just covered themselves by making additional release. And this worked perfectly well. The population of rabbit crashed drastically in just a few years. Bang, they were all dying. But suddenly, as you can see, boom, 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 it went up, as you can expect it. So what's the reason? Well, simply that the virus select among the rabbit those that were resistant. It's a simple co-evaluation process. The virus select the host, but then the host being resistant will select the viruses. And so especially when you target a species like a rabbit that replicate like mad, only few subjects can repopulate the system. And very, very interesting, and this was something difficult to explain, but I'll try. So let's imagine that at the very beginning, you have in your lab a line of rabbit. So genetically stable, you have a stock of rabbit that you use as a reference for sensitivity to the disease. And you have your strain that you will release into the field, right? So this is your inoculum and your rabbit that you use in your lab to test sensitivity. You start with a virus that is highly virulent. You put it on your rabbit into your lab, they all go down. You go in the field, you release myxomatosis as a biocontrol agent. You take your car, you drive 100 kilometers, you stop in the countryside, and you wait for the virus to reach you, right? So the virus will, from your initial site, will transmit from rabbit to rabbit to rabbit, etc. Of course, during this journey, the virus will be submitted to some evolution, to some pressure of selection. And what you will observe when you obtain you're 100 kilometers from the releasing point, you collect the virus when it reaches you, and when you will go back to your lab and look at the virulence of this virus, you will realize that you have select mild virulence. So the virus that you will collect at 100 kilometers from your releasing point will be less virulent than the one that you release. Simply because throughout the cycle of transmission, you will select a virus that has the right compromise between virulence and transmission. And if you're too virulent and shoot your host like a bullet in the head, you don't transmit efficiently. So this is what the story is telling us. So that the higher virulence is not obviously the most fitted virus in terms of evolution. Now, think about what I just showed you. This one here. So the pathogen evolve, but the host will evolve. So when your virus has circulated and become endemic, when it's coming on the population, the second round is exposed to hosts that are more resistant. And there, obviously, if your virulence is not high enough, you're not able to infect. And so what you will observe is the opposite. When the resistance of the host increase by the natural selection, then you will see that the highly virulent strains will pass. But globally, you will still have less mortality induced by the virus. But the situation will completely change in terms of evolution. At the beginning, you select less virulent, and once you build up the resistance of the host by natural selection, then only the virulent strains will be able to transmit. So that's very, very important to keep in mind in a model, is that according to the resistance of your host, you select at the first site less virulent strain, while later, when the host increases its resistance, you will select more virulent. And it's one of the things that we observe with Marek disease, for example. If you analyze the virulence of the strains according to time, it's quite clear that we have constantly select more and more virulent, including by exposing a resistance conferred by vaccination. So in the 50, myxoma, then 90 finished, no effect, we back, the rabbit is back in business. Then they say, well, let's try another disease. And they move to a calicivirus, rabbit hemorrhagic disease virus. And as we can expect, very efficient effect at the beginning. And then nowadays, the rabbit are back in business because it's the same story going on and on and on. Okie doke. Hans, maybe we should do a short break here. And after we move to my favorite subject, the biocontrol of carp. So let's have a CHV3 as a biocontrol measure against carp. This is again an Australian story. So common carp, as I mentioned, is a fish very important in aquaculture. It's actually the third most important fish in aquaculture worldwide. And it's not a natural species in Australia. It has been brought there, again, by human to develop aquaculture. In the 70s, they had a massive flooding in Australia. So carp escaped from the farm and colonized the rivers. And the rivers were already highly polluted at that time. And carp, being a very strong fish, they were well adapted to this polluted environment, and they proliferate like mad. Rabbits proliferate rapidly, but the carp, a female of few kilo, can make few millions of spring per year. So it's really highly prolific animal. And then observing these carp becoming dominant in the ecosystem, the Australian, as they used to do, thought about a way to kill the carp in the ecosystem. And some scientists came with the idea to use cyprinid herpes virus 3, so the virus that different labs are studying, but I'm one of them. And they sought to produce this virus in large stock, very, very large stock, and then to release it into the rivers, right? So this was the idea. So I initially thought that this was just an hypothesis. And then at the end of 2016, 2017, I was contacted by an editor of Science, and she informed me that they were planning to release the virus the next year. And this is called the National Carp Control Plan. Really very important. I don't remember the cost of this research program, but it was huge. And so the editor of Science asked me a simple question. Do you think that this process will be safe and efficacious? So I said, well, we have to analyze the case. And so at that time, we were studying the impact of the environment on this disease, because as I mentioned, it's highly influenced by temperature. And looking at the temperature that you found in Australia, most of Australian river, it was quite obvious that this virus will not be efficacious because you're not in the range of that kill the fish. And also you have important gradient of temperature. And so my interpretation was that fish will express behavioral fever and will save their life. And so we built the case, but it was very, very simple and rational argument, like selection of resistant. What will happen if you have few percent resistant, but the next year it's colonized by resistant animal and so on. And this was end up to be a letter in Science that created me some trouble. But and this is I promise to give advice to the young generation. I already mentioned, do not believe papers. Go to the data, look at the data yourself, be critics. It's not a right, it's a duty as a scientist. Don't swallow what is written in paper. And if I have the chance to retire, one day I will write a book on epistemology, which is the heart of designing an experiment and presenting your data in an honest way. And this paper will for sure will deserve a chapter. And this is a very important paper that was published by these guys that were also involved in the program. So you cannot exclude potential conflict of interest. And this guy wanted to address two simple questions related to safety and efficacy. The safety is, will the virus that we plan to release kill native species? That's obvious. If you release the virus into the rivers, kill the carp, but kill also the other fish that you want to save, that's not useful. So they did a simple experiment. They went into rivers, catch fish, and expose these fish to the virus to determine if this virus was safe for non-target species. On the other hand, you want to know if your virus is efficacious, meaning that will kill the different genetic background of carp that you will find in the ecosystem. So here again, what you do is simple. You go in the wild, you catch the carp, you bring them in your lab, and you expose them to the virus. You expose them how? Well, obviously, with conditions that mimic what will happen in the wild. So not an IP injection, because you're not going along the river and say, oh, we want to kill you, come here. We will make IP injection for free. No, you will expose your carp by bath, what we call bath, meaning releasing the virus into the water. So let's see their data. That's a very impressive table. Let me explain this. So on the top, you have the percentage of mortality. So meaning the number, the fraction of fish that die after exposure. And in bracket, very, very important, the number of animals tested. So 50% means nothing unless you know on how many animals it has been tested. And then below you have two subdivisions, CG3 challenge and the control. It's important, of course, to realize that your fish in the control don't die and to see the effect. If you have mortality in your negative, then you have a problem. And then you have two, for each column, you have IP, which is not highly relevant. It could be used as a positive control, but this is not what you will do. So you should favor bath, and this is the column. Now let's see the results. So all the red here are the carp, Cyprinus carpio, so the species that you want to kill. Okay, what you see, oh, very interesting. It doesn't kill everything. Even by some carp in some place seems to be resistant. Only 60%, 63% of carp that die after RP injection. Okay, then funny stuff like a group of 1 or 2 animals. That's top science. 2. How can you put in a table a results based on 2 animals? 1, yeah, you cannot go below. Huh? So that's quite impressive. So and for example, here 100%, and this is used in the results and the discussion. Oh, they die by 100%. Yeah, but you had only 5, and you had no control group, no real group with bass. So that's unbelievable. So these guys claim on their data that the carp from different regions in Australia are sensitive to this virus. 63% after IP, 50 by BAS. What about killing 50% of the carp when each will generate millions of offspring the next year? Now, what about the other species? Well, these are 3 species that are not carp. So this is the same table. I'm just highlighting different species to help you. And you see here, even by BAS, the virus is killing them. Here too, here too. And when you look at the conclusion of the abstract, there were no clinical sign, mortalities, or histological evidence consisting with a viral infection in wide taxonomic range of non-target species. And this is one of the bit that support the file that has been submitted to the Australian government to support the release of this virus. It's clear like this. If you don't believe me, read it. You have the reference. It's just one of the piece of the puzzles. And this is science. This is built by scientists. It has been reviewed in a journal, Journal of Fish Disease. It's a pretty good journal. It's not PNAS or Cellulose and Micro, but it's a good journal. So don't believe what is written. It's your right, but even more, it's your duty to control and criticize even what is published. Because there are people that publish and make conclusions that are opposite to the results. I'm very proud of me because I managed to describe the story without being upset, which is not the case most of the time. You see, this is becoming mature. Mature. Okay. Now, very briefly, two other aspects of viruses to kill target species. And in this case, I'm going to talk about bacteriophage as a way to kill bacteria in an infected host. So basically, you want to use bacteriophage to kill non-self cells, the bacteria, into an infected organism. And you know, the problem of antibiotic resistance in bacteria is becoming really a huge problem. My son is now a surgeon, and this is a nightmare for him. You have a lot of patients that die in hospital from multi-resistance antibiotics. And for sure, there is a discrepancy between the emergence of multi-resistance bacteria that go much, much faster than the discovery of new antibiotics. And this is becoming really, really a problem. So a long time ago, and actually in 1919, some scientists thought to use bacteriophage, which are a very efficient way to kill bacteria, as a therapeutic against bacteria. The idea is simple, just like a biocontrol agent. If you have a phage able to kill the pathogenic bacteria, then you administrate the phage to the patient, and this phage will act as an antibiotic, finding the bacteria and killing them. And there are, of course, many advantages of this principle. The first is that in theory, it must be more difficult for bacteria to become resistant to a phage infection. I'm not a bacteriologist, so I'm not sure it's true, but there are certainly mechanisms by which the virus could select a resistant bacteria. An obvious interesting point is that your phage will multiply into the bacteria. So the more bacteria are present, the more your phage will multiply, and antibiotics don't multiply. So that's an advantage in favor of the phage. It's certainly very, very specific to the bacteria. The probability that the phage is able to infect on one of our keratic cells is extremely low. And this is demonstrated by experiment. They have a high therapeutic index, meaning that you can put a huge dose of phage and it doesn't seem to be toxic for animals. And more importantly, when people in clinics describe the problem of antibiotic resistance, is very frequently associated with biofilms. And it seems that at least in surgery, the biofilms is a terrible problem. So this biofilm, you know that the bacteria live in a kind of matrix surrounded by this extracellular polymeric substance that will not only protect them from the immune system, but also from the access of different molecules. and this makes them highly resistant. And so people thought that the bacteriophage could be a very interesting way to treat the biofilm, because by definition you have a concentration of bacteria there, and so when you concentrate the host, it promotes the spread of an infectious agent of the host. So this makes sense to use them as an alternative to the antibiotics. And so, of course, this looks like an advertisement to promote the science in in bacteriophage. I received this slide from one of my colleagues working on this topic, but for sure, the concentration of the bacteria could promote the spread of the phage into this biofilm. Some bacteriophage also, they induce or they synthesize proteins that degrade the extracellular matrix. And so by inducing this infection, we could disrupt the biofilm. And finally, and this is, I think, the most interesting aspect, if bacteria become latent or dormant, because that's a problem of antibiotic treatment. You treat with the antibiotic, everything goes well, and then a few weeks after, it takes over again because few bacteria were dormant into this biofilm. And this phage could be an approach that to keep dormant, like the bacteria, the phage, and then react immediately when the bacteria become active again. So as I mentioned, the first trial were in 1919, when Felix Derel used it to treat bacteria dysentery. Surprisingly, this approach was stopped in Europe. In Soviet Union, they were quite developed, and there are currently some product. And nowadays in Europe, the interest start again, and there are clinical trials that are going on now. And I hope that this will solve the problem. But it's an obvious biocontrol measure. Viruses to fight against cancer cells. So here again, the idea is to kill cells inside an organism, and the cells are self cells which are abnormal. So the idea is to use really a virus that will be specific of the cancer cells, and will kill cancer cells but not normal cells around. If you have this special tool, then it acts as like a surgeon at the level of individual cells. Now, again, the idea is not very recent. The first attempt to treat a carcinoma of the cervix was done with attenuated rabies virus. And you see that we had a lot of trials until very recently, 2016, where the first viral oncolytic treatment was approved for melanoma in the United States, with quite variable results, but some of them are very impressive. There are people that had metastasis, and this virus catch the metastasis. Now, something that is very impressive with this viral oncolysis, so not all patients react positively, but in patients that react positively, it is quite clear that you must have indirect effect of the viral oncolysis. The viral oncolysis does not explain everything, because you don't put enough virus into a patient to catch all the infected cells. So this led to the idea that the virus was infecting and killing some of the tumor cells, but then was able to launch an immune response that was not able to develop without the virus. And so the mechanism is as follows. This is a rationale of how it works. The first is, of course, this safety measure. You need to have a virus that discriminates healthy cells from tumor cells and act only on the tumor cells. When replicating into these cells, it will lyse the cells, which will release the virions. And again, you can think about an amplification phenomenon. But very importantly, this virus will most probably, by inducing innate immune pathway of the cells that detect the viral infection, will induce an inflammation. And so the virus most probably trigger some kind of inflammation adjuvant effect that the tumor tried to prevent. And you will release antigen. And so all this create this what's called immunogenic cell death ICD, where the virus induces the death of the cell in a way that will be immunogenic, will lead to immune response. And you probably know that when the cell dies, for example, from apoptosis, you will promote cross-MHC class 1 presentation, which is excellent to induce cytotoxic T cells. So that's a fascinating topic at the border between the virology and immunology, and certainly a really great field. Now, what is, I think, a chance for this old concept of viral oncolysis, 1990, it's a very, very long time ago. I think that nowadays we have such a knowledge of the different pathway at the different level, at the surface of the cell, and the inflammation molecules, cytosolic nuclear mechanism, that we can really design a virus that will have the perfect property, and perhaps stupid things like viruses, for example, acquire tricks to prevent PKR activation into a cell. If you make a virus that has a defect in this inhibition, then when it will go into the cell, it will switch on these phenomena and make the cancer cell being pro-inflammatory, something that it cannot express, otherwise it will be eradicated from the body. So I think that's it's a very complex field, but for sure I'm pretty sure that the viruses will create a gap into this process. Again, there are plenty of different viruses families. Each virologist tries to push his virus against the other, and especially when you tackle such a diverse entity like a tumor, because that's a problem of tumor. You have a huge diversity even within a tumor, and so very likely it's not one virus that will solve the problem, but a collection of viruses. Also, come back to this this picture of Michelangelo. What is fascinating nowadays is that you can really, based on the fundamental knowledge that you have of an immune response, you can build a virus that will really induce the immune response that you want to develop. And for example, there are this TVEC, which has been approved by the FDA. The virus has been genetically modified so that they will express a GMC CSF, and so which is a very stimulating cytokine. And so when the virus is killing a cell by replicating into the cell, it's not only providing the antigen, but it's also providing all the accessory stimuli to make this antigen immunogenic and inducing an immune response that the tumor has been inhibiting sometimes for months or for years. So that's really fascinating field. And if you look at now the number of clinical trials that are going on, the number increase every day, and it's clearly a great hope for patients who are suffering from cancers. Last point, viruses as substrate of vaccine development. Well, of course, and there are plenty of different vaccines that are derived from the use of viruses. And the very first is the subunit vaccine. I already mentioned this baculovirus from GSK. It's used, we use virus to produce a protein that is now incorporated into a subunit vaccine. So you should already be convinced by this example. There are plenty of heterologous vaccine against viruses. What means heterologous? Remember, it's when you use two pathogens, well, two viruses that are antigenically related. One being benign, causing a benign infection or no disease, and the other one being lethal. An example is the very first story I told you during the inaugural lecture. If you take the orthopox virus genus, you find here variola major, which killed human, and you have vaccinia virus, that is a related virus infecting cow, and this causes a benign infection in human. Because these viruses are phylogenetically related and have in common highly conserved antigens, once you are immunized by vaccinia, you are in fact protected against all the orthopox viruses. So this is called heterologous vaccination. You use one viral species to protect against another phylogenetic related viral species. Another example frequently described in veterinary vaccinology is the use of shop fibroma virus, which is a lepropox virus related to myxoma, but which in contrast to myxoma induce benign lesions in rabbits. So that's another example of the use of viruses to develop vaccines. So subunit vaccines, heterologous vaccines, and then recombinant virus. Recombinant vaccine, and the term recombinant in vaccinology is confusing because it can be used in different ways. A virus can be recombinant. Recombinant means that during the process of production, you use recombination to induce a genetic modification. And the genetic modification can be the remove of a gene, a deletion, can be also used to insert a gene into a transgene into a virus. And I already described this story about the production of a vaccinia virus for oral vaccination of fox against rabies. So the idea was originated from Jeffrey Smith, who was my boss in Oxford, and he was one of the first guys to prove that you can make a recombinant virus expressing a foreign gene. And then this idea was used by Jean-Pierre Lecoq, who took the genome of the rabies virus, make a cDNA because it's RNA virus again, so use this reverse transcriptase, makes a cDNA and then clone the open reading frame coding for the glycoprotein G of rabies into the genome of vaccinia virus strain. So the very same virus that was used to vaccinate human against smallpox. And this virus is a GMO. It's a genetically modified organism. And when this virus replicates in two cells, it replicates just like the wild type. You cannot differentiate the viruses in cell culture, but the difference is in terms of number of particles produced. But there is a major difference, is that this recombinant virus will express, in addition to its own protein, the glycoprotein G of the rabies. And so when you inoculate this vaccinia virus, recombinant virus to a fox, the fox will produce the glycoprotein G and will be immunized. And I described you the story about this vaccine, which is very unique. It's a vaccine that consists, it's a snack for foxes. So you have this little plastic bag that contains infectious vaccinia virus, the recombinant virus, and this is surrounded by fat and by this croquette, which is meat made with a rotten chicken head. So they were collecting this from a slaughterhouse, and it has to mature to smell like mad, because it's very important that the final bait is very smell, so that the fox find them very quickly in the wood. And I describe you how they are released in the wood. And this turned out to be very efficient and led to this eradication of rabies into Europe, which was quite a major achievement. Now, this man was my boss, and Professor Pastore was a great human being. I'm very impressed and really sad to realize how fast people forget you when you disappear. Most of my students completely ignore the existence of this man, and some of my colleagues in the faculty too, which is very sad, because if a man like him does not leave trace, does not leave memories in the faculty, then all of us will disappear the day we leave. So I found this extremely shocking, and so I had the idea to develop a piece of art in our faculty that will stay there and will force students to remember him. And this is my advice to the young generation. First one, never hesitate to honor your master or your PhD supervisor, and that at the end it's always good. And don't wait that the guy is dead to honor him, because unless you are a believer, I think it's everybody, as I said, in the inaugural lecture, every single person needs positive feedback from time to time. And yeah, promoters of thesis did not arrive on hers just to receive complaint. If they are human beings, it's for some of them it's difficult to detect, but they need positive feedback. So I thought and my second advice is that when you want to do something, even if it's unlikely that you will succeed, try and try hard. And so I always admire that I travel a lot in the United States, and when I was working in San Diego, I was very impressed by an artist called Georgia Gerber. I don't know if you know her. She makes a bronze sculpture of animals. It's bloody expensive. And they are in the White House, they are everywhere. And so one day I was thinking about Paul Pierre, and I thought it would be great to have a Georgia Gerber sculpture illustrating the work of my master. And so I contact her in the middle of the night. I wrote an email and I say, this is my story. I like this man, he passed away. And she replied, she said, well, let's do the project together. And so that's the project, and it will be a lunch in the spring. We'll have a big stone with a rhabdovirus here and a fox, and this magnification of the stuff. And this is the sculpture. They went sent from California to Liège a few months ago. This is you recognize, huh? It's a beautiful rhabdovirus. It has been printed, 3D printed with wax, and then they make it in bronze. And this is the fox. It's about that big. So I will invite you on for the inauguration this spring. Yeah. So that's my two advices. Last bit, recombinant attenuated vaccine against CHV3. I already described this. It's one of the major achievements of our lab over the last few years. We did this recombinant viruses, and there is something that I'd like to stress. And you know, we live in a society where the decisions are not always rational in terms of science. And I'd like to explain you something very simple that you don't need to be at the university to understand. And I'm asking a question: how do we produce an attenuated vaccine? And to make a long story short, there are two different ways. So at the beginning, you have always a wild type virus. A virus, and I'll illustrate this with mice. I don't like mice, I already told you, but from time to time, it's good to recycle old slides. So you start with the wild type virus, and this wild type virus, when you administrate it to the target species, it kills them. Now, how you produce an attenuated vaccine from this? And there are two approaches that I like to call the random approach, which I don't like, and the smart approach that I prefer. So the random approach is the approach that has been used for decades and for some of them centuries. So what you do is that you take this virus and then you will start by generating diversity. You pass your virus a large number of times, and obviously genes that are not essential for the virus to grow in cell culture will acquire mutation. And so if you watch carefully, I symbolize three genes here, the red, the green, and the blue. And you see that in this bulk, in this soup of virus, you start to have diversity. One has lost the green gene, etc., etc. But you don't control this. You don't control the genetic determinants. It could be a point mutation. It can be an entire piece of DNA which is removed. We don't know. It's completely blank. Then once you have generated this diversity, you cannot, of course, administrate a vaccine with this a mixture of different genotypes. So you need to go through a cloning diversity procedure. So you will clone this variant, then expand them to obtain pure population of these different variants. And then finally, you test them in vivo. And what you're looking for is a virus that is still able to replicate in cell culture, but doesn't make the mice sick when you administrate it. Spend me 10 minutes to change the eye here. See, this one is alive, but not feeling well enough. This one is with the eye open. So here, when you end up with these viruses, if it's a new virus, you know that you transform a virus from a wild type to something, wild type fully virulent to something which is no longer pathogenic. You still have to control it to confer protection. But if you don't have basic knowledge on the pathogenesis, even if you sequence these two viruses, you will detect here a lot of mutation, but you don't know which one confer the attenuated phenotype. So it's a kind of artisanal way to produce a virus. Now, the smart approach will consist to build your strategy on studies on the pathogenesis. For example, you know that the green, the red, and the blue genes are virulence genes that the virus need to complete and induce pathogenesis. And what you do, you take your scissors of molecular biologists and you cut nicely these genes. So you remove completely these genes. So reversion is impossible. And there you obtain the perfect virus. Now, there is something very important with these two different approaches, is that the right one is considered as GMO. So genetically modified organism is a technical definition. So it defined as a genetic modification, which is the product of human hand. If you pass the virus here, mutation comes from the sky, it's not a GMO. If you go this way, it's a GMO. What's the point? Well, the point is that regulatory registration of the vaccine, or even the possibility to bring a virus into a vaccine into a country, is sometimes defined by GMO. Some countries, for example, does not, like Japan, does not want to listen about GMO. So if you make the perfect vaccine here by deletion, pure clean deletion of the gene is refused. If you obtain this gene, this deletion, point mutation by random passage, it's accepted. So this doesn't make sense. Just to make sure you get the point, if I take this, let's say we have one gene, which is a virulent gene, essentially. You make a point mutation here, which is acquired by chain of passing the virus. That's accepted. It's accepted. While the risk of reversion is huge. Huh? If you have one point mutation in one way, it could be the other way. You take the gene, you remove completely the gene, that's a GMO, no risk of reversion, and it's forbidden in some countries. And even more stupid, if you think that you find a virus you go in the river, you pick up a strain and you find a strain that is deleted for the gene, you make exactly the same modification by molecular biology, then you become a GMO while the other one is not. And that's the society in which we live. And it's the consequence against of some dogmatic people that present GMO like, well, GMO, they are not good and bad. They depends on a case-per-case basis. But educating a society with thinking is something that most people don't like. It's much more easy to educate people black and white and say GMOs are against, and I will protect you from GMOs. But with these kind of typical law that go above a technical concept rather than on case-per-case situation, scientists like me who produce GMO viruses, which are extremely good, extremely safe, are embarrassed to register the vaccine into some countries. While these vaccines are much, much safer than the classical ones, that's for sure. You don't need to have a degree in a PhD to understand. Now, I will finish with some of the studies that we did with this live attenuated vaccine. I'd like to stress the role that Maxime played. It was my assistant and a very bright PhD student. And this is one for the first study that we published on this paper, but there are a few that came out after. Now, the concept, the vaccine that we developed for this virus is compatible with what we call mass vaccination. Max, yeah, this was the first name of the guy. I didn't realize that. So it's mass vaccination. And the idea was to be able to vaccinate thousands of fish simultaneously. Because if you have to vaccinate small carp of 1.5 centimeter, one by one, forget it, it's impossible. So the idea was to make a virus that will be released into the water. And that's why we investigate the portal of entry and the studies that I showed you earlier. Of course, with a vaccine, and especially a live recombinant vaccine, you need to address safety and efficacy. And I already showed you data of efficacy last time, so we'll skip this very, very quickly, where I show you that we vaccinate with wild type, well, with the vaccine, and then challenge with this wild type virus expressing luciferase. I showed you the data last time that were showing that once the fish are vaccinated, the challenging material can even not invade the fish. You have no light emitted by the vaccinated fish, suggesting that this vaccine confer sterile immunity. I show you this. Now, what about the safety? And the safety is frequently an issue that come into competition with the efficacy. If you think about the safety of an attenuated vaccine, the rules are very clear in terms of registration. Your vaccine must be extremely unable to spread from a vaccinated fish to cohabitant, which is the opposite of efficacy. If I was a farmer, a fish farmer, I would like to vaccinate one fish or two fish, then release them into the lake and ask them to vaccinate all their friends in a safe manner. No, this is not allowed by regulation. You must have a kind of control of the spread of your vaccine. So this is one aspect of the safety. And this is, I think, beauty in veterinary virology and in veterinary in general, is that we have a more holistic view of a system than human medicine. We're not centrated to one subject. We actually create an entire group, and we talk about herd immunity, etc., etc. And so when you talk about the safety of an attenuated vaccine for fish in the context of mass vaccination, you have to address the safety for the vaccinated subject. So with in other words, the spread of the vaccine within the vaccinated subject, but you have also to investigate the spread of the vaccine from the vaccinated subject to the cohabitant. And this is what I will show you here. So to address this, again, we use this great technique, which is bioluminescent imaging, and we produce in a comparable manner for the wild type virus and the vaccine equivalent strains that were expressing luciferase. So the Luc cassette is inserted both in the wild type and in the vaccine in the very same place, which is a non-coding intergenic region. And the idea is to use luciferase to reveal the spread of the wild type or the vaccine within infected fish, just like I explained last time. And this to address the safety within the vaccinated fish, but also later as a mechanism to study the spread of the vaccine from vaccinated fish to cohabitant. And so the experiment that we did here is very simple. You inoculate your fish by bath, so you just drop the virus into the water, just like a normal vaccination procedure, either with the wild type virus expressing luciferase or the vaccine strain expressing luciferase. Then we collect these fish at the indicating time point. The experiment is unfortunately for the fish shorter for the wild type virus because the fish die, while with the vaccine you can prolong this experiment. And what we did, and I think this is very, very important if you have to design an experiment, is never ever rely on a single readout. Because if your readout introduce a bias or give you artifact, all your study rely on artifacts. While if you rely on different techniques, which rely on completely different approach, it's impossible that systematically these techniques give you the wrong information. And this is why in this very large experiment, every single fish were analyzed by IVIS, by qPCR, and by histology. Of course, not for all the organs, because you cannot do it. But you see that gills, for example, were analyzed by all the three readouts. And if you look this, if you read this paper, a very important thing throughout the paper is that for as a defined time point and a viral genotype, the same symbol means the same fish. So you can go throughout the paper and correlate the data that we obtained by IVIS, qPCR, and histology. So you can really see that the readout gives you the same same result. So let's see the results of the IVIS. That's quite a complex figure, a lot of work. I will explain it. So horizontally, you have the different organ analyzed: the gill, the heart, the skin, kidney, gut, liver, and the spleen. And vertically, the left represent the results of obtained with the wild-type virus expressing luciferase, and on the right, you have the results obtained with the vaccine expressing luciferase. Then it's a very nice way to present the data. You have every single symbol represent one fish, and as I mentioned, you take the symbol here, go here, you will find the results for the same fish. And then the gray bar represent the number of positive fish among the 6 fish analyzed per time point. So you have a kind of general view of the data, but if you want to zoom in and see individual data, you can trace them into the figures. So obviously, if you look at the gray bars, it's the easiest way to analyze the data. You see that the wild type, as we know, all the fish are positive at day 2 on the skin. They remain positive until they die. And this is due to the fact that the skin is the portal of entry. You also see that the signal builds up in terms of flux time. All the other organs become very rapidly positive, and on day 4, all the fish are positive for nearly all the organs. If you look at the vaccine strain, let's start with the skin. It's very interesting. All the fish are positive on day 2, just like for the wild type, but then the signal does not build up like we observe for the wild type, and it's sustained for a couple of days and then it disappears. Now, about the other organ, you see that they turn positive at some stage, but it takes some time. So the vaccine strain is basically recapitulate the replication cycle of the wild type, but he's doing it at the slow motion rate and at the intensity, which is much lower. You see that, for example, the gut, the liver, you have a transient expression for some animals, but even not all of them. So it's really the deletion that we made into this virus really impaired the replication cycle of the virus. Let's zoom of the skin and the gill. You see that the vaccine infection is lower, delayed, and shorter. One thing that I like also in this story is that the vaccine is actually further demonstrating that the skin is the major portal of entry. If you go back to this slide, you see here that they're all positive for the skin, while the gills, for example, most of them are already positive. So one could claim that the gills and the skin are parallel portal of entry, and that we see more fish positive for the skin because simply a higher replication cycle. What is nice here is that it showed that while all the fish are positive for the skin with the vaccine, the gills will turn positive only around day 6, which means that it demonstrates that the gills is a secondary site of infection and not an alternative site of entry. So this, I like it very much. qPCR, does it correlate with the data that we obtained by IVIS? Well, yes, otherwise I will not present it. What we observe is that, again, we have a lower, delayed, and shorter time of expression of the virus, replication of the virus into this organ. So the data of IVIS perfectly correlate with the qPCR analysis. What about histopathology? Well, very same. So that's what was done by Maxime again. I don't know. Well, I know you have a very good pathologist here in Ghent. In Liège, this experiment was quite large, and Maxime was one assistant in the pathology department, designed a grading system of the lesions, and then they educate 3 scientists to recognize these 3 grades of lesion, and then they give the slide in a blind manner of all the fish to these 3 scientists and ask them to score the lesion 3 times randomly. Then we made mean of the scoring, etc. So we managed to make statistic out of these anatomopathologic lesions, which are quite obvious. And you see the lamellae here, the fusion. It's quite, when you are educated to recognize them, it's very easy. And this turned out again to correlate the data of the IVIS. We observed that the wild type virus was inducing very, very nasty lesions, and the vaccine only grade number 1 and then vanished. So altogether, this qPCR, IVIS, and histology confirmed that our vaccine was safe at the level of the individual. Now, what about the spreading of the virus from one vaccinated subject to cohabitant? And this is typically what I like. The science I'm doing on fishes is a little bit like Pasteur science. It's simple. I like simple things. And you take a tank, tubes, a fish, you connect tanks or not. And so here we did a very simple experiment. We took some fish, the black fish, and we inoculate them by bath, mimicking a vaccination, either with the wild type look or the vaccine look, right? So you take this fish, two separate tanks. One, you vaccinate them with the vaccine expressing look, the other one, wild type expressing look. And after, we will mix them with cohabitant sentinel fish. That is my graph will be the white fish. And these are the fish that we will analyze by IVIS to determine whether or not they were contaminated by the vaccinated or the wild type of fish. And we designed two very simple systems that I nickname water sharing and tank sharing. In the water sharing, the virus that were initially vaccinated or inoculated, the black one, are upstream of a grid where you find downstream the naive sentinel. And these are the fish that we will analyze from time to time to determine whether they were contaminated by these fish. And in the tank sharing, we mix the black fish and the white fish. So this question is, how do you recognize the fish that were vaccinated and not? And this is called fin clipping. You just make a small incision in the fin so that you can recognize the fish that is a black fish. Tank sharing, so obviously here the virus can only be transmitted through the water acting as an abiotic vector, while here the transmission can occur through the water, but also through kissing, touching, skin-to-skin contact, or whatever the interaction that the fish express. So let's see the results. So difficult figure, the little circle represent the wild type virus, the square represent the vaccine. So let's see first the results of the water sharing, so just transmission through the water. It's quite impressive. All the rectangles are below the cutoff. So that means that not a single sentinel fish was detected positive by IVIS. In other words, these fish vaccinated here probably secrete some virus, but the amount of virus secreted is so low that it's not sufficient to contaminate the fish downstream. So no transmission through the water acting as an abiotic factor. What about tank sharing? All the little dots here represent the wild type. You see massive contamination of the white fish, like here. And here, only two fish, the two square here, this is the mean, the white one, the two fish that were transiently positive at day 10 and 18. So this virus is really inefficient. This vaccine is inefficient in spreading from one fish to the other, which is extremely good for the registration and the safety of this vaccine. So again, this was a lot of experiment, and Maxime Pinghu Yang was a PhD student from China. She's now back in China as professor. Megan Ronsman and all the team involved a lot of energy. And you know, common carp is produced mainly in Asia and particularly in China. So China represent 80%, so 80% of the world production. So it's very important to bring your vaccine to the market in China. And we're very pleased that Ningbo Sancheng Biotechnological Technology has acquired all the rights for vaccine in China. This was signed last year in the middle of the COVID crisis. And we have a very intense and interesting collaboration with this company. And I'm really pleased. I'm looking forward to go and visit them. But importantly, as you probably know, traveling to China is not fun at the moment. So that's all the different items I wanted to tell you today. So I hope I convinced you that there are a lot of substrate. Viruses are an important source of substrate of biotechnological innovation. I just described some of them, but there are more than that I described, and it's a fascinating field. So this was my last lecture. Huh? It's to be honest, I'm pleased. It's because I'm quite busy too, and when you have to take 1 or 2 days out of the week to prepare something, well, it's good, but it has to stop. And I'd like to thank again Hans. As I mentioned at the very beginning, receiving an award like the Francqui share is prestigious for sure. But knowing that he proposed my name means really, really a lot to me. Really, yeah. It's and I'd like also to thank. Well, as you detect, I'm quite an emotional person, so I will not talk too much. But I really think that doing science is a privilege, and for the young generation, you should see science like this. If you don't feel that science is something important for you, if you don't love it, then don't do it. There is no room in science to be successful if you don't love it. And doing science is a great privilege, but being a scientist gives you also a lot of duty, and you should be honest and make sure that you deserve to have this privilege and to be one member that defends this duty and the importance of science. I'm also asking you to be honest in science for sure, but to be a scientist that is integrated into science, into the society. Our society has a bad view on science, and I don't know why, but they tend to trust more stupid guy on Facebook and the internet than real scientists, and it could be that we don't spend enough time explaining what we're doing and the rationale. I still hope that the dogmatic position of some people is our fault because we didn't spend enough time explaining. I have some doubt, to be honest, but please keep trying and be happy in science and enjoy science. Thank you very much.
Once more, an excellent presentation. Of course, there are always some questions. I have a few, but I'll.
I was just wondering how carp are doing now in Australia.
So far, so good, because they didn't release the virus. They're doing well. Carp is an opportunistic fish, which is favored by bad quality of water. They're so strong that they can survive in a drain, and so because the Australian government is, well, they're building a lot of dams for agriculture. As a consequence, there is a bad condition, and this favor these strong, resistant fish compared to the native species that are much more sensitive and require good ecosystem. So the population didn't change. They replicate like mad.
They're not going away soon.
No. And they're not only strong, but they're extremely prolific. When you generate 1 to 10 million offspring per individual per year, it's
the rats of the market.
Yeah. Well, it's more than rats. It's millions. 10 to the 6. It's yeah.
So does it mean that the national carp control plan was cancelled?
I don't know if it's cancelled, but they stopped the release of the plant so far. So the virus was in theory released end of 2018, and it has not been released so far. And key people at the head of this program have been looking for a job. Yeah.
I have also a question, like concerning because you still see that the vaccine after application there is still, of course, low level. Okay.
Yeah.
Are you planning to study the invasion mechanism? Because if you could have an additional, there should be a mechanism behind it, how they get into it. If you could also block that one, then you have only a superficial.
Yeah.
Or do you think that the immunity will be extremely low, then
Yeah.
you will have no protection.
Yeah, that's it. The blocking, that's one of the many deletions that we tested, and we have deletions that have an even more drastic effect on the replication of the virus in vivo that completely block it. But then you have extremely safe, beautiful, cannot be safer, but you have no efficacy at all. And we actually, Ian Zhang is a PhD student in my lab, is studying the ortholog of this gene into angulate herpes virus 1. And there the situation is even more interesting. You have a virus that is not affected at all in cell culture. You replicate like the wild type, and you put it in the ill, and it goes away. So we think that this protein is involved in some kind of blockage of an innate immune mechanism, which is probably more developed in ill than in CAR. Yeah.
Also because you still have replication in the skin.
Yep.
What about bacterial superinfection?
Well,
no,
it's you don't see the lesions. That's the IVIS is so sensitive that you see it, but you clinically you don't see lesions. No, well, depends. I'm sure that if you use the IVIS to drive your biopsy, then you will find it. But if you do it randomly, you don't see it. It's really, you can barely see sometime at day 2, 3, if you know. But in blind tests, people cannot differentiate the mock from the vaccinated group. Yeah, extremely safe.
That's not a student, that's a professor, so that's why.
Yeah, that's
He's upstairs.
yeah, that's good. It looks okay, huh? Yeah, that's good. Good spirit. Wake up every morning like a student.
On the 2A side, you mentioned the ribosomal skipping, but also the ribosomal read-through and the
Yeah.
falling off.
Yeah.
Do you know what the percentages are of these different processes?
This is a study in the paper I mentioned. It's about the dropping off is about 60%. So that means that in terms of stoichiometry, if you analyze by Western blot, for example, you will always find a majority of your front sequence. Yeah. So it's in our system we see it. The bioluminescent, which is the front one, is highly expressed, but the GFP is much lower than what you will observe if you make the same construction, putting GFP alone in the first position. Yeah. So that's the weakness of the system. Well, there are several weaknesses. The first is that you leave a tag to your front and you add that one amino acid to that. If it's report a protein, it's okay. But if you want to study the biological property of position number 1 or 2, and then the stoichiometry of your 2 separate protein or the fusion protein, it's likely to be variable from according to the sequence that you cloned.
We inserted a few cleavage sites.
Yeah.
After the 2A, so it leaves off the 2A peptide.
Yeah.
You just have coding.
Yeah. At the ahead and 5' of the second one. Yeah. And which one did you use? Which T2A that you used? Okay. Well, it's a nice system. I'm using it, and what is beautiful in our case is that you can, because that's the weakness of the IVIS and the confocal is that you don't have a link between the two. You see something macroscopically with the IVIS, then you take a biopsy and you go to the confocal and you have to make an immunochemistry. But here you take this very same sample and like a prey bird, you watch with IVIS, take your sample and go to the light sheet to the confocal microscopy and you see the cell that generate the signal. Yeah, it's very elegant. Yeah. So if you need this construction, we can pass them. Just send me an email.
By post.
Yeah. Dry on a piece of paper and then we send it. 1 Diet Coke for the plasma.
Yeah. Okay. I think we can stop here. I want to thank you all. once more for
Thanks, and
the exhaustive list of
enjoy
talks
science.
that I think we will put, if we're allowed to, put the talks also on for the students, because the students don't want to.
They're all yours.
Everything is now taped, and we will fire it to them, and I will ask them one question just to see, and mark on if they have plenty of it. Sorry, but
Selection.
but stimulated to have a question. They can learn a lot about that. So
Yeah.
very peaceful. Also, my young students, there were a lot of them always complaining that I like you to say, shut up, it's a privilege to be one of the students that can do research, and that's always a kind of it's difficult to get it in their brains, but it's a joy of life instead of kind of suffering.
But they should complain too.
They should complain, yes.
Every day, yeah. That's the rule.
You did it also as a student of work.
No, I think I see. Oh yeah. Yeah. There was
You had to work too hard.
no. At university it was okay, but at high school it was terrible.
Okay.
Terrible. I remember. I was fired a couple of times. Yeah. It's not a joke.
So your parents were not that happy.
Yeah, no. But I think it's good because.
So you ended up to become a veterinarian because.
No, but not this. But I remember the school where I went, and when I met my wife, was the consequence of being rejected from another school.
Okay.
So if you're not rejected from one school, you don't end up.
You're
Then never ever will meet my wife.
So
It's terrible.
she's happy.
No, I don't know. You have to ask her. You see her tonight. You will ask. But on my side, yes. I'm the most happy husband in the galaxy. No problem.
I will tell her.
No, yeah, you can tell her.
Okay, thank you. We'll stop here.
It's probably recorded.
It's recorded. I will. Yeah, I'll send it.
I knew it. This is why I said. Thank you.
Thank you. Hello.
Yeah.
Thanks.
You finish.
Yeah.
Is there something wrong with the thing?
No, it's done. They did it. They even sent the letters to the Minister of Finance.
The medal.
No, they don't do so anymore.
Because I remember that they.
Yeah.
Because I've seen that they sometimes also give the medal to the people.
No. This is the international Francqui share.
Yeah, because I asked them. They said
No.
we don't come. We don't come. They don't come. They don't come.
Ik heb de 17e mijn doctoraatsverdediging.
Let me know.
Ah ja?
Mijn publieke
Ja. Public
defensie.
Ja. Je mocht afzetten.
Ik vroeg me af, wordt er nog een link
Also,
opgezet? Een videolink?
you tell me where we have to meet and then.
Dat kan natuurlijk, dat is like dat hij dat wil. Hè.
Ik zou het graag hebben als het mogelijk is.
Ja, tuurlijk kan
Waar
dat.
mensen in het buitenland zijn die ook kunnen volgen.
Ja, oké. Ga je dat ik op mail zetten?
Ja.
Dan kan ik die link aanmaken en dan kun je dat rondsturen hè, naar de mensen dat hij wil. Is er...
Het is de bedoeling dat iedereen die link krijgt. Of ook hebben ook in zelf.
Ja, tuurlijk. Maar ja, de dag van vandaag kun je moeilijk nog anders. Hè.
Ja, er zijn een paar mensen in Spanje die
Ja.
zo over het weer komen voor auto spreken.
Ja.
Zover is het nog niet.
Nee. En zeker niet met de dure brandstof tegenwoordig. Hè.
Het is 1,6
Ja, fuck vind. Waanzinnig.
Het is voor 2 jaar geleden was het 3 jaar geleden was het 0,9.
Verder dubbel vind.
Ik herinner me nog een keer op reis geweest naar Griekenland. 5 jaar geleden of zo. Daar was ik reis 2 jaar.
Ja?
En je zag op straat, al die auto's zijn vol met stof.
Ja, natuurlijk. Daar herinner je mee. Zeg nog iets anders. Is er gesproken over een boekje of zo? Of hoe zit dat? Voor je doctoraat? Ik weet dat niet, hè?
De herregels.
Alsjeblieft?
De herregels.
Maar ja, als je je tekst hebt, laten we dat in boekjesvorm maken. Hè. Dus laten we uw cover ontwerpen. Als jij een idee hebt, stuur dat naar de ontwerper. En toen moet je vragen, Hans, hoeveel boekjes moeten er zijn? Die Die zaten allemaal. Moet nog geregeld worden.
Single cell RNA is approved.
Wanneer is uw interne verdediging?
Vandaag.
Geweest. Komt ervan. Oké. Dus ik vermoed dat er minor corrections of...
Corrections tegen afgesproken 18e. Stuur ik het door naar de members of the jury.
De 18e.
18e. Zelfs een week.
Challenging for school.
Ja, als je nog wilt printen, moet je nog... Ga je in de miserie komen. Hè? Ja, hij had al praktisch 4 dagen nodig. Zeker 4 dagen voor hem te kunnen printen. Dus als je hem maar een week hebt voor aan het deur stuurt, wanneer gaat ze antwoorden? Want je kunt helemaal laten...
2, 3 dagen. Wel is gewoon bekijken. Nee, ja, meteen leven zijn niet doen. Ik heb nog veel comments gehad toekomen. Misschien zou het wel vroeger kunnen. Maar met mijn vriendin was dat eigenlijk 2 dagen vooraan. Dus we gehad.
So what's the plan?
Dat was ook tijdens corona. Misschien dat iets betekent.
Ja, normaal moet je rekenen op 4 dagen.
Maar ik wist niet dat dat via onze dienst ging. Ik dacht dat dat via een centrale UGent-service was.
Je kunt dat ook. Je kunt dat ook.
Maar ik wist niet dat er verschillende mogelijkheden
Ja. Oh, als het via de centrale dingen gaat, heb je weinig ruimte voor de cover. Oké, op zich is dat ook niet zo erg. Ik ga een keer vragen.
Dirk, thank you very much.
Good luck.
The key of my car.
Oei, quite important.
Is het een huiscar?
It's a professor's car, so it will not be that.
So now the
Yeah, it's always, you should have one place where you put always your keys. Gewoon die put op toon.
Ze haalt te veel.
Ja, ofwel, rond te veel zeker. Ja. I think this is coming from the, wat onder dat weer?